And our next speaker is Professor Erik Halgren. Erik received his PhD from UCLA in 1976 and stayed finally as an associate professor until 1997. And I think it was mid-1990s that you visited Helsinki and did some interesting experiments with Vita and also with Tommi Rai, who is in the audience. And then 1997, Erik moved to University of Utah, where a new MEC center, even with two MECs next to each other, was set up. And I have actually many fond memories from those times because we visited Utah many times for a couple of weeks, because the prototype of the vector view was installed there before it actually functioned very well, so that we did the final phases of product development basically at the University of Utah. And I remember Erik was using the very early version of FreeSurfer and some kind of precursor of M&E software, and he was singing there, not in the rain, but something like, "I am inflating the brain, I am happy again." And then in 2000 he moved here to Boston to set up our wonderful MEC laboratory at MCH, and then finally in the middle of the first decade of this millennium, he moved back to home, I would say, to California, to UCSD, to become a professor of radiology there. And I think it's fair to say that Erik is very keen in understanding how the invasive measures from the brain and the surface measures that we collect with the EEG and MEC are related, and how the different stories that they are telling us about the brain relate. And I think his talk will touch some of these issues, and his title is Stages of Word Understanding. Please.
Thank you very much, Matti. That was a lovely introduction, and you know the dates better of my life than I do, and I would have expected you to. So I'm honored to be amongst this group. I'm honored to be here to celebrate David's 40th anniversary, the sole author of the sole publication that invented this wonderful technology. And I started working with intracranial recordings, and when I started working, it was, of course, anything field potentials were so we did units, and then any idea that you could do something outside the head was ridiculous. And eventually, through MEG, I was converted to the possibility for discovering something important. And it's very nice to be here amongst you. And I want to thank the previous speakers for having such wonderful talks and keeping everybody in the room. So I'm going to try to move quickly. I'm sure everybody has saturated LTP. I'm going to talk about stages of word understanding, and the first part of the talk is going to be phenomenological, descriptive. What are those stages? What do they do? Where are they located? When do they occur? What is their neural basis? How do they interact? Who do you see them in? And it is wonderful to be phenomenological. I like that. But the big payoff, of course, is to try to understand something about these fundamental issues in word understanding, these fundamental issues that psychologists have been working on a long time, but they've been trying to do it with a black box. And we can look inside that box a little bit. We can shake it. So one question I'm going to broach is acoustophonetic processing, and it took a long time to figure out that was the right word, but there are people here who will disagree, I'm sure, affected by the lexicosemantic context. That just means, is it, yeah, in the front row too, in my reviewer. Is it is the expectation we have for what a word means, does that affect how we hear words? For what word you're going to get based on the context, is that affecting how you hear things? Secondly, do written words have to be recoded phonologically before lexical access? So, in other words, if you're reading something, do you have to kind of sound it out inside your brain? Maybe you don't actually hear it, but you have to sound it out inside your Wernicke's area or something to access what word it is and what it means. And then the third question is, does lexical access precede semantic encoding? And, a neuroscientist would say, yeah, well, duh, you have to know what the word is before you figure out what it means. But, maybe that's not so. So the brain's actually, we have to respect it. And I was invited by the organizers to talk about, MEG and intracranial, and I'm going to mix them up ad libitum. So it actually the story starts with EEG. This is from Marta Kudis and Steve Hilliard's publication in Science, these many years ago, and they showed normal subjects these sentences visually, and then the last word would be normal here in the dark line. It was his first day at work, or sometimes it was he spread the warm bread with socks. And when they spread the warm bread with socks, they got this dashed line up here, this N400, 400 milliseconds after the word onset, there was this peak. It was recorded like right here. It's where you record it relative to the ears. And they had as a control, she put on her high-heeled shoes, and shoes was big font, and that evoked this P3. So we just forget about that. So you can actually get this N400 in lots of different situations. If like you prime it with a sentence, fine, but you can also prime it just with a preceding word, cat, then dog. So if you compare the response to dog and jet, well, it's much bigger to jet because you've decreased it to dog. So the priming decreases the N400, at least that's how I see it. And this prime can be, it can be in the auditory modality, even though this is visual, or vice versa. It can be a picture. It can be, some inference. So this is like really a very semantic sort of influence on this. And that kind of, this idea that you evoke an isolated word will evoke a great big N4, a normal, big-sized N4. So that gives you the idea that what must be happening is that something's deciding, some antecedent process is deciding this is possibly a word, and then it's emitted the N400, and then you have some sort of modulation that will decrease it if it's easier to figure out what the word is, if it's easier to do this lexicosemantic encoding. And again, that was a long fight, and I'm sure I'll get some feedback on that. But so this is kind of brought out into contrast with the observation that you can compare Blardo, which is, yes, it's not a word, but it might be a word. And so the difference between Blardo and this consonant strings is like, between a congruent and incongruent real word. So in other words, to a pronounceable non-word, you get to just define N4. Does that mean the N400 doesn't have anything to do with language? I don't think so, because what it's saying is that we don't know yet that it's not a word. It's the process of the brain figuring out if it's a word, accessing the meaning, trying to figure out what meaning it is, that is the N400. So it has the implication also that our brains use the same process for learning words as for understanding words. It suggests that, and I think this is the case. So I have David's picture here of the areas that are activated, greater activation in bold to sublexical stimuli, the nonspecific controls. And so this is, we could say that acoustophonetic encoding, and I have the microphone, I'm sorry. And then there is another figure here from Binder that is the areas that with bold was showing differences related to semantics. And so this is suggesting that maybe the initial the initial processing is focal, and the semantic processing is slightly distributed. And I'm so thankful to you for doing this summary slides, because I could never read these hundreds, thousands of fMRI studies. And it's very gratifying to know that the F word has not been used much today. So in the visual modality, this relates to the visual word form area. And this is, again, bold studies that radiological convention, it's on the wrong side. But where various contrasts between words and consonants here, and in this case between words and sensory controls, produce activation in this posterior fusiform gyrus. And lesions in this area, this is one of the typical small lesions, can produce an alexia, difficulty reading. So you also see with MEG, this is a study we did with Spanish, English bilinguals, and you get, again, this sort of left hemisphere activation back in this region when people are reading words. And that's at about, this was really early, I understand why, but around 150 milliseconds. So, So we did a study where we compared real words to consonant strings, and to false fonts, right, which were just made by cutting up the consonant strings and then cutting up the letters and then pasting them back together. And we reasoned that if there was a letter form area, that would be shown by some difference between the consonants and false fonts without a difference to the real words, and that the difference between the real words and consonants, which is seen for the word form area, that would show that. That contrast would show the word form area. And so in the orange here was the fMRI, and that's showing this contrast between real words and consonants, and this is showing in red the contrast with fMRI between consonants and false fonts, so letter form, word form, in this ventral stream. And it's consistent with other work like by Vinquier and others that there are a series of specialized areas down here that are coding for more and more complicated aspects of words. So with MEG, if you do an estimated strength in these different areas, you can see that at about 160 milliseconds, the activity estimated here is distinguishing between up here are words and consonants, and down here are false fonts. And that later, here's 225 milliseconds, you get all three being distinguished. So that's nice, but the inverse problem is ill-posed. So every time somebody shows you an inverse solution, you have to remember that they aren't sure, and different inverse solutions give different results. So I always like to use some intracranial recordings to try and figure this out. And so we recorded, this is just one subject, and I hope it's in the corner, you can see it. And in this subject, we did fMRI before they were implanted, and we got electrodes right over the area that was activated by this contrast between consonants and false fonts. And lateral and medial, we got no activity, and right above this, we got a lot of activity to these stimuli. And here is the local field potential, average local field potential, and here are the high gamma power. And I don't know if you can see it because it's not projecting all that well, but you see this peak here? That's the false fonts, and this peak is the consonants. Uh-oh, it's bigger to the false fonts. Well, you can't tell with LFPs if it's excitatory or inhibitory, right? So we say it's bigger, but you can't say just because it's bigger that you have more activation, you have more activity. And in fact, if you look at the consonants, here, they're much bigger than the false fonts, recorded simultaneously by the same electrodes, just treated differently. This high gamma power, we just take the high gamma and we, average it from 70 to 190 hertz or something. So this is just the noise of the crowd. This is the chanters, okay? It's like you're in a football stadium. There's a little group of people who are chanting, right? They are spatially organized, and they are temporally synchronous. And that is what the LFPs are saying. That's what MEG is saying, because MEG is just, the LFPs recorded outside the head. And LFPs are actually generated by currents, and this current source density identifies where they are. All of these have the same influence. They are a subsample of the activity. And some people say, you want to hear the people who are chanting, maybe it's the orchestra, rather than the audience. We don't know, but it's a subsample. It's some special people, some special neurons, some special synapses, as opposed to these other measurements. And fMRI PET, as was pointed out, very highly correlated with high gamma power, which is just a reflection of single unit activity and fast synaptic activity. So you lose, going from here, you lose information encoding. You don't have, the differences between all these different neurons in the little area that are each saying something different. When you go from here to here, you lose temporal resolution. High gamma power has great temporal resolution, millisecond temporal resolution, just like MEG. So here you know from this, that this activation here with fMRI has this time course. So this is saying, yeah, there is also LFP generated there at the same time. So when I got back from Rita's lab, and thank you very much for your hospitality, it was exceptional, we started the study with the same task that was described here with the words. And this was in Utah, and we were looking for the N400, and here's like 10 subjects. And here are the fields that were recorded. And these fields have, just a nice dipolar pattern, I guess, and locates right here. And this was previously done in Rita's lab by Rita Salminen. And this sort of, Wernicke's area single dipole had been reported. And we we're interested in using Anders' technique, which was this anatomically constrained minimum norm estimate, developed originally, based on Mati's discovery of that. And we see here this subject, when we do this MNE type, or it's called DSPM in our lab, we get something that, yes, involves this Wernicke's area, but also some Broca's area and some anteroventral temporal cortex. So what is right? Again, what is right is indeterminate from the MEG. You have to go inside the head. And we've recorded from thousands of places, and we've been doing it for a long time. And this was a summary we made of where the generators were of the N400 prior to ever recording MEG. And so we thought that the fact that we're getting, with DSPM something up here and something down here as well as something up here made us like that method. And that's what we're going to use here instead of the dipole method. I don't know. I think it's right because it is so bad. It is so smeared. It maybe is a good method. So just this then leads to some sort of model where in comes visual information. Then there are these series of steps, and then one of these steps is a key spot here is kind of a bottleneck of the P170 that's encoding the word form, and then around here at N400, which seems to be maximal here in the base of the temporal lobe. So we looked then with this method at the phase locking values that Lachaud introduced, which is just how consistent the phase is in a given frequency between trials. And so this is a phase locking with the trial onset of these different phases, well, actually, between the phase locking is between different locations, but at different times after trial onset. All right. So this is a plot of the absolute phase locking values, and I don't know, people in the audience who've used them might be shocked at these. These are like 0.7 level of phase locking that is evoked in this situation here. So that's, usually it's much, much lower. So this is like almost, they're almost saying exactly the same thing in these frequency ranges. And so this is between this location, which is there in the letter area, and all of these other areas, which can be quite distant here in the prefrontal cortex, for example. That is, though, at a lower threshold, which I can't see here. I think it was cut off, but it's more like 0.3 or 0.4, which is still very high. Okay, so there is this first sweep, and then there is a broad, wide band up to about 30 hertz phase locking between the areas. So initial sweep and then phase lock. And that is to words. And of course, it's much smaller to these other, to the consonants or the false fonts. And it actually, in the same recording, when we got very medial, here over lingual gyrus, this is a blow-up of the bottom of the brain here. And here we are in this area that's responding to words as opposed to false fonts. So this is the high gamma response to the word. Here's the high gamma response to false fonts. We have an area that's responding to the false fonts in the lingual gyrus. So, actually, if you look all over the brain, there's a lot more response to false fonts than there is to words. And this is actually consistent with a lot of EEG and MEG data. And that kind of makes you wonder, like, did the brain really evolve to operate on false fonts? I don't think so. But that leads one to an interesting observation, which I'm going to show you here. And that is, when you present a word, there is a very strong phase locking between these two areas. When you present, and these are coming in random order, right? And when you present a false font, there is no phase locking between these two areas, even though this area is like super active. What's going on? Maybe the way things work is that there are non-specific streams, and these non-specific streams are automatically activated unless the template is matched. And if the template is matched, these other streams are inhibited. And this sort of thought can also be applied to the N400. So this is communication in order to integrate, and this is communication in order to focus. Because the thing is, you have to realize, and this is the beauty of MEG, and this is something you never think about with fMRI, is it has to evolve in time. You don't know anything until you know it. This is, you have to bootstrap your understanding over time. And this is an example where you don't know yet if it's a word or a false font. As you learn to know that, then you have to redirect on the fly what you're doing with it. And that's maybe what we're seeing here. Okay. So next is, what is their neural basis and how did they interact? And I'm going to go fairly quickly through this. We are fortunate to have, in a very small number of cases, microelectrode array recordings that we can relate to the language studies. And this is a neuroport, which has 96 contacts that go 1 millimeter into the brain, and we can separate out many single units and call them pyramidal cells or interneurons. And we also have something called a laminar electrode, which has 24 contacts in 3 millimeters, and with that we can deduce current source density, and from current source density and the multi-unit recordings, you can deduce where the sinks and sources are, which is the active synapses, what layers they're in during the task with respect to time. And that's important because the brain is a pizza. It's about the size of a pizza and about the thickness of a pizza, and it's layered. And the organization, the way it's wired, depends on these layers. So if you can figure out what the inputs and outputs are as far as the layers go, you can figure out a lot about the connectivity. So the way the connectivity works is that the first input is into layer 4, like in this top-down way, bottom-up way rather, like going from V1 down to wherever. And that's mainly an AMPA GABA-A operation. And then this activation goes up to upper layers and then down to lower layers, and they go back up, and the associative input is coming in up here. And that's more of an NMDA GABA-B operation. So these are two separate systems. They're about equally strong. They have as many fibers underlying them, and they're just as organized. But they are probably doing very different things, and we saw there seems to be in language processing a first pass, which is a feed-forward passing through, which is some sort of template matching, and then the second pass, which is broad, associative, and hand-waving. And that is this going up here into the upper layers. And that indeed is what we see when we do the laminar recordings. This is the a task that Anders did, which was a size judgment task. People saw single words, and they decided if the word represented an object or an animal that was bigger than a foot in length, if it fit in a shoebox. And this is half the words repeated. This is to the new words, and this is the difference between the new and the old words. And this is 185 milliseconds at the time, maybe, of this activation down here of the ventral word form area. And this is the time of the N400. And you can see in the new minus old, there's nothing here, and there's a lot here, including the side of the electrode, which is about right there. So this little microelectrode, the first thing that happens is around 200 milliseconds, and you get a sync here in the middle layer. That's consistent with this feed-forward template matching sort of operation. And then up here, the time of the N400, you have a sync in the upper layers, which is consistent with being associative. And then you can see also there is some oscillations there, which we'll get back to. And those oscillations are more apparent if you superimpose single sweeps. And so we superimpose the single sweeps here, and what happens is you have this activity that appears disorganized here, but actually has a lot of theta rhythm in it, and it is theta delta. And then the stimulus comes on, and you have a sharp response, which is the feed forward, which is not present in the baseline activity, and then it resets the theta, which is then what you see here in these successive cycles. And that's what all this is about. And so here's a single sweep. And this is the position of the pyramidal cell, and this is the middle layer. And so these are spontaneous thetas, which are showing this alternating sync between upper and middle layers. The stimulus comes on, it's reset, and then you have these alternating syncs again. And so the hypothesis is that this initial sync is acquiring information, and then the upper sync is associating it. So recently, Ksenia Marinkovic and colleagues in our group this has implemented one of the methods here that of source space theta localization. And she was comparing these pseudo words, these pronounceable non-words, with real words, and getting a very big response, which you see between the red and the green here, in the language areas to this difference. So post N400, this event-related theta is differentiating meaning. So this continued activation, and it makes sense, you figure out in the first pass, in the N400, that it's not a word, and after that, you're done. But you continue if it is a real word. And I'm not going to go into that because I'm not going to have time, but we have some evidence, again, Marinkovich, that this later activity is related to reprocessing, which you do get to jokes. What do you call a cow with its feet in the air? Ground beef. So the N400 is really killed by that, right? There's no N400. This is his feet are in the air, you know, he's a beef, you know, that's a no N400, but great big activity later because you have to reprocess it. All right. So how do they develop and who do they occur in? So I'm now going to go back and give a little more information. Here we don't study infants with intracranial recordings. A lot of them are done, but they're usually really, really sick. So this is something which we can do is infants in the MEG. And we decided, my graduate student, Katie Travis, who recently graduated, decided to look at whether what their N400 was like. And they're like, 14 to 18 months old, and she used two tasks. One was she presented sounds that were matched to words. So obviously all auditory stimuli, and so cat, and then there was this noise, but it was a really good noise because it was noise vocoded. So we took different bands and we matched the envelope of the power in the different bands. So it should have been a very good control. And they were individually matched to each word. And we made sure that our averages consisted of the words and their individually matched controls. And lo and behold, the babies had a beautiful N400 that was peaking in the same sensors and at the same time as as humans. And spoken as a father. And then what she did was she showed a picture of a cat, and she played either the word cat or she played some other word. She showed a picture of a ball, and she played the word cat. So she compared those, and again, you're comparing exactly the same word. There's no question that maybe some of the differences are due to sensory level differences, and I feel your pain there, David, because this is what we obsess about too with auditory stimuli, and we found, and she found that again there was a response that looked very much like this incongruity response to the incongruous words, the words that didn't match the picture in the babies that was very much like you see in adults. And so this is the incongruous minus congruous DSPM map in the infants, and this is in the adults. And you really can't tell the difference. And this is the response to the words and the response to the noise. And again, you have this left frontotemporal response. So what does this mean? It means we can study it in babies. They're 14 to 18 months old, and maybe, as you were saying, that it's going to be possible to do something clinically important. To me, it's really interesting because to me it means that maybe babies are learning words in a way that's as much top-down as bottom-up. That they aren't just doing it through some sort of massive exposure of millions of words over time, that they abstract from repeated sensory presentations more and more abstract representations. I'm sure that's going on. But at the same time, there's something coming from the most cognitive levels of the brain that is abstract and is organizing this learning. Another special group that we're not going to get in the epilepsy unit is native deaf signers, and so we have been studying them. This is Farian Ramirez and Leonard and Mayberry, and we find again that there is very similar responses to incongruous versus congruous words in the deaf, the native deaf, and in the hearing, even though their auditory cortex is not been used for audition during their life, and they don't show any early response in their auditory cortex of visual stimuli. There's no evidence for rewiring. It seems to be really that this cortex is used for that. This cortex is used for lexicosemantic encoding. And whether you are using signs or using visual words or using auditory words, it's still being done in the same way. Okay, now we can go to these fundamental issues, and I have 12 minutes. So, the first question, is acoustophonetic processing affected by the context? So, one typical effect that has been around for a while is the Ganong effect, and so that is you're more likely to hear a D if it's presented within the word duck than if it's presented within the word desh, right? So the lexical context is affecting your perception, your reported perception. You have to decide, what am I hearing? And so that has led to a series of models by some of the earliest models of the PDP models of Ellman and McClelland. And those models have arrows going both ways between the words and the letters, right? They are interactive models. That's very controversial, though. Norris, that's been cut off here, I'm sorry, has, among others, but in Norris's merge model, there is unidirectional influence, and the bidirectional influence is occurring in a separate structure. And you can reproduce everything doing that. And maybe, you ask somebody what you hear, maybe that's a different process than actually, processing speech online. So Katie and Matt designed this study where, again, you see a picture, you hear the word, cat, picture, word, or you hear cat, but after the cat, or you hear a noise that matches cat. So this comparison is going to tell you the acoustophonemic processing, and this comparison is going to give you the lexicosemantic. And you pre-present this picture, so the process is, the context is there before, so you'd expect that maybe the incongruity effect should start at the same time as the processing. But in fact, it's 120 milliseconds later. Long time. And here's an individual sensor, our favorite sensor here above, I don't know, Wernicke's area. And you can see between words and noise, you get a big difference. And that starts at about 60 milliseconds. And between congruous and incongruous words, they're right on top of each other until here. And then they start differentiating way out here. So, and then these are just the maps. So that's interesting, and maybe supports one type of psychological model. And then we went inside the brain to try and see if we could get supportive evidence. And this is from a grid, big grid, and a microgrid. The microgrid spaced at 1 millimeter. These are spaced at 1 centimeter. And we're looking here at the high gamma power and the local field potentials. And it just seems to be that this early difference between words and noise, which is on this side at about 100 milliseconds, is in different places often than the later difference between congruous and incongruous words, even at a very small scale. So maybe there is also a spatial differentiation early. And the problem with this experiment, even though David and other people have done studies many people have done studies showing that about this latency around 100 milliseconds or less, there is phonetic, different phonemes are if you move back and forth, the voice onset time, blah, blah, you get differences in this time domain. we wanted to verify that, and so we looked at this word-noise contrast, and we compared that to a ANOVA that looked at the variation between different consonants in real words. And this consonant difference was happening in the same places and about the same time as the word-noise difference. So that makes us feel better that we're really looking at the early parts of word processing, of word understanding. Now I'm going to talk about this just a little bit. This is where we somehow, this patient was slated for an anterior temporal lobectomy, and before the lobectomy we were able to record from a electrode right here in the posterior margin of the resection. And this is like 96 contacts and recorded about 120 units and plus, and under many, many different tasks. And this is a inhibitory interneuron. This is an excitatory cell. You know, they're blah, blah, blah. It's marvelous technology. And these cells are responding specifically to words. They don't respond to tones. They don't respond to, you know, clocks or staplers or harps or whatever. And they respond less to backwards and forwards words. And they do show this word-noise response at the same time that they're showing this very high level of discrimination between different phonemes or between different words. These cells are responding to particular words and to particular phonemes. And so we're a little cut off here, but this is consistent, given this location, with the model that has been proposed by Rutschecker, and that is that language words are understood through this dorsal stream going forward from Heschel's gyrus instead of the classical idea that they're understood by going backwards to the planum and around, you know, supramarginal gyrus and the arcuate fasciculus forward. That's used for articulation and for maybe, you know, spatial location or something, but that these are being used for understanding which speech it is, which word it is, and then so we have a model that would go like this. Well, I'm about to change to orange, and I'm going to talk about this next problem of, do written words have to be phonologically recoded before lexical access? Wait a minute, am I orange or am I yellow? Red. Am I red? Am I at 40? I thought it was green there for a while. All right. Well, I'll talk about that during the. I'll talk about these last two questions if people want to hear about them. 3 minutes? Okay, I will do it. So basically, phonological recoding, I explained to you what that is, and there is a lot of psychological literature. One part of that psychological literature says that we recode words automatically, but that we don't use that for understanding what word it is unless we are a poor reader or it's a difficult word. All right? And that's from Seidenberg a long time ago. And you'll notice this is the same thing that I was showing you from Anders, that by 185 milliseconds, you are getting some activation up here, we imagine, to written words. So Ksenia, again, Marinkovich did a study where she looked at the N400 with Meg to heard words versus read words, and it seems like it's the same N400. So that would be consistent with this this idea that in order to access the word, you have to go up into the meaning of the word to a visual. You have to go up here. You have to go to the dorsal part. Now, this I'll just say, in fact, we find that there are modality-specific N400s, and I can't go into this slide more except to say there are modality-specific N400s, and so that is inconsistent with this obligatory phonological recoding. But this is the kicker. This is the really exciting result, which is that if you show an in two of these units in this, this recording, we're responding to both visual and auditory words. And the responses to individual sounds of letters and to individual written letters were correlated. All right? So that is really phonological recoding. And that shows what you can find out with single units that you could never find out with any other method. So you understand what this is? So you see a word, a letter, and you hear the sound of that letter within words, and they are highly correlated. And the time of this seems to start at a time when this is R prime using a moving window starting at 175, was able to significantly classify, was significantly correlated between the modalities. So that's probably early enough to contribute to phonological recoding, to understanding a word from its sound when you read it. So this is related to the earlier work that Apostolos showed, and here we're decoding, Alex Chan is decoding which word you're being presented, or whether it's an object or an animal, based upon a machine learning paradigm, and this is with MEG. And we were able to replicate that with intracranial results, and I'm going to show you one, and that is that starting at 200 milliseconds in first pass processing in the ventral stream here, in anteroventral temporal lobe, you're discriminating between words and objects in unit activity or current source density. And what this means is that in this first pass template, you know that the word refers to, a written word refers to an object or an animal, probably quite a bit before you know what word it is. So semantics are not after lexical identification. Thank you.
Time for questions.
I'm wondering if you'd be willing to speculate on what might be going wrong when things go wrong, like in kids with SLI or individuals who have a stroke. Is it a timing thing? Is it a coherence thing? There's so many possible explanations based on what you showed today.
Well, I would imagine that there's lots of things that can go wrong. I mean, it's pretty amazing that we're able to do it at all, right? I mean, that we can see these tiny differences in either auditory or sensory stimuli, and within, a few 300 milliseconds, pick out the word out of thousands of possibilities. So, and integrate it with all our knowledge. So that is, it's amazing what we can do. From no matter what the IQ, people can still do it. So I think this is a very complex system, and probably in different people, different things go wrong. And maybe ultimately what we can contribute as brain imagers is methods to distinguish different things that can go wrong.
Yes, Ray.
Hi. For the work that Katie has been doing, I was just wondering, are you doing head movement compensation on the infants? And if not, then how do you deal with that?
Katie sits in the room with the babies. We aren't doing it. I mean, we've tried. It just didn't really. We didn't need to. We didn't have to, and then it just seemed complicated and more effort than it was worth. So Katie sits in there with a baby, and she says, "Now," and the recording says, "Look," and it comes on, and she doesn't. She starts the trial by pressing a button when she sees that the baby is ready and is looking, and then it's presented, and the baby's looking because it's interesting, and then the look, and then the word, and so they're packed in really well. And so that seems to work good enough.
Okay.
I'm curious, just as a model, if I hear cat, and I haven't yet recognized the word. I haven't even finished analyzing the sounds or the letters. How do the cat features become activated such that they perceive that I then recognize the word?
I mean, how?
See, since you concluded with saying that the semantics actually are at least at the same time or precede the access to the word form, I guess I'd like to hear how that actually works.
Well, so
not so obvious how that might
That's
work.
not so obvious, is it? No, but the here's my thought. I mean, we've only seen that with visual. With auditory, we don't have these same laminar recordings, okay, that have shown the first pass processing. So I'm not sure it happens with auditory. That's another problem. But you can see these cells in the anterior superior temporal gyrus are responding to individual words long before you know what word it is. So it's just, they are pattern separation machines, right? So you get a stimulus, and they are discriminable based on sounds somehow, and so they discriminate them. So I don't know beyond that, because the actual point in time when you can distinguish between cat and catch maybe hasn't been met. So how do you do that? Then you're just dealing with probabilities and how those propagate through. We don't know that. In the visual system, you have all of the information. And I would argue that there is no difference in principle between recognizing something visually as an animal or an object and recognizing words that refer to objects and animals. Objects maybe have more similarity between them. I don't really know that they do. A hammer and a chair and a car, those are pretty different, right? And, a snake and a giraffe. So we don't have any trouble with the concept that we have these visual processors that can distinguish between different objects by their appearance, but we somehow have a problem with imagining that can't work for words too. Maybe we have, this direct pathway through the visual stream that's recognizing words as objects, as visual stimuli, and that you take this whole set of visual stimuli that match, objectness, and they're getting a feed forward, right, it passes in two streams. They're going in parallel and they're converging up there. One stream is going through the object and animal areas and are making an early discrimination, right, between them, based on what it looks like, and the meaning, and the other part is going and meeting back at in the front, So with just some information about the shape of the word, of the written word. So that is, an associative convergence causing long-term potentiation. So I think we have to have our minds open to that happening, that we can recognize the meaning of words based directly on their appearance rather than their lexical, their lemma, right?
All right, I think.
Can I have a very quick question?
Should we take a vote?
Yes, you can.
Just building on David's question, I was curious, one way of looking at this semantics preceding lexical access, what about frequency effects? So if you could actually modulate semantic activation based on the frequency of the word, you could predict whether the semantics precedes lexical access or not.
Right. Well, frequency effects are very interesting, and I think they are very early. Frequency effects are very early, and so are like frequency of bigrams and trigrams and all of that. So I think that it's all the same thing. I think it is. There are very early effects in the visual stream. We're designed for that, I think. But maybe I'm wrong. I think the first pass is very strong.
All right. Time to thank the speaker again. very keen in understanding how the invasive measures from the brain and the surface measures that we collect with the EEG and MEG are related, and how the different stories that they are telling us about the brain relate. And I think his talk will touch some of these issues, and his title is Stages of Word Understanding. Please.
Thank you very much, Matti. That was a lovely introduction, and you know the dates better of my life than I do, and I would have expected you to. So I'm honored to be amongst this group. I'm honored to be here to celebrate David's 40th anniversary, the sole author of the sole publication that invented this wonderful technology. And I started working with intracranial recordings, and when I started working, it was, of course, anything field potentials were so we did units, and then any idea that you could do something outside the head was ridiculous. And eventually, through MEG, I was converted to the possibility for discovering something important. And it's very nice to be here amongst you. And I want to thank the previous speakers for having such wonderful talks and keeping everybody in the room. So I'm going to try to move quickly. I'm sure everybody has saturated LTP. I'm going to talk about stages of word understanding, and the first part of the talk is going to be phenomenological, descriptive. What are those stages? What do they do? Where are they located? When do they occur? What is their neural basis? How do they interact? Who do you see them in? And it is.
And our next speaker is Professor Erik Halgren. Erik received his PhD from UCLA in 1976 and stayed finally as an associate professor until 1997. And I think it was mid-1990s that you visited Helsinki and did some interesting experiments with Riitta and also with Tommi Rai, who is in the audience. And then 1997, Erik moved to University of Utah, where a new MEG center, even with two MEGs next to each other, was set up. And I have actually many fond memories from those times because we visited Utah many times for a couple of weeks, because the prototype of the vector view was installed there before it actually functioned very well, so that we did the final phases of product development basically at the University of Utah. And I remember Erik was using the very early version of FreeSurfer and some kind of precursor of M&E software, and he was singing there, not in the rain, but something like, I am inflating the brain, I am happy again. And then in 2000 he moved here to Boston to set up our wonderful MEG laboratory at MGH, and then finally in the middle of the first decade of this millennium he moved back to home, I would say, to California, to UCSD, to become a professor of radiology there. And I think it's fair to say that Erik is
publication in Science, these many years ago, and they showed normal subjects these sentences visually, and then the last word would be normal here in the dark line. It was his first day at work, or sometimes it was he spread the warm bread with socks. And when they spread the warm bread with socks, they got this dashed line up here, this N400, 400 milliseconds after the word onset, there was this peak. It was recorded, it's like right here. It's where you record it relative to the ears. And they had as a control, she put on her high-heeled shoes, and shoes was big font, and that evoked this P3. So we just forget about that. So you can actually get this N400 in lots of different situations. If like you prime it with a sentence, fine, but you can also prime it just with a preceding word, cat, then dog. So if you compare the response to dog and jet, well, it's much bigger to jet because you've decreased it to dog. So the priming decreases the N400, at least that's how I see it. And this prime can be, it can be in the auditory modality, even though this is visual, or vice versa. It can be a picture. It can be, some inference. So this is like really a very semantic sort of influence on this. And that kind of, this idea that you evoke, an isolated word will evoke a great big N4, a normal, big-sized N4. So that gives you the idea that what must be happening is that something's deciding, some antecedent process is deciding this is possibly a word, and then it's emitted the N400, and then you have some sort of modulation that will decrease it if it's easier to figure out what the word is, if it's easier to do this lexicosemantic encoding. And again, that was wonderful to be phenomenological. I like that. But the big payoff, of course, is to try to understand something about these fundamental issues in word understanding, these fundamental issues that psychologists have been working on a long time, but they've been trying to do it with a black box. And we can look inside that box a little bit. We can shake it. So one question I'm going to broach is acoustophonetic processing, and it took a long time to figure out that was the right word, but there are people here who will disagree, I'm sure, affected by the lexicosemantic context. That just means, is it, yeah, in the front row too, in my reviewer. Is the expectation we have for what a word means, does that affect how we hear words? For what word you're going to get based on the context, is that affecting how you hear things? Secondly, do written words have to be recoded phonologically before lexical access? So, in other words, if you're reading something, do you have to kind of sound it out inside your brain? Maybe you don't actually hear it, but you have to sound it out inside your Wernicke's area or something to access what word it is and what it means. And then the third question is, does lexical access precede semantic encoding? And, a neuroscientist would say, yeah, well, duh, you have to know what the word is before you figure out what it means. But, maybe that's not so. So the brain's actually, we have to respect it. And I was invited by the organizers to talk about, MEG and intracranial, and I'm going to mix them up ad libitum. So it actually starts with, the story starts with EEG. This is from Marta Kudis and Steve Hilliard's been a long fight, and I'm sure I'll get some feedback on that. But so this is kind of brought out into contrast with the observation that, you can compare blardo, which is, yes, it's not a word, but it might be a word. And so the difference between blardo and this consonant strings is like, between a congruent and incongruent real word. So in other words, to a pronounceable non-word, you get to just define N4. Does that mean the N400 doesn't have anything to do with language? I don't think so, because what it's saying is that we don't know yet that it's not a word. It's the process of the brain figuring out if it's a word, accessing the meaning, trying to figure out what meaning it is, that is the N400. So it has the implication also that we basically, our brains use the same process for learning words as for understanding words. It suggests that, and I think this is the case. So I have David's picture here of the areas that are activated, greater activation in bold to sublexical stimuli, the nonspecific controls. And so this is, we could say that acoustophonetic encoding, and I have the microphone, I'm sorry. And then there is another figure here from Binder that is the areas that with bold was showing differences related to semantics. And so this is suggesting that maybe the initial The initial processing is focal, and the semantic processing is slightly distributed. And I'm so thankful to you for doing this summary slides, because I could never read these hundreds, thousands of fMRI studies. And it's very gratifying to know that the F word has not been